This was chapter 42 of Marx, which is interrelationships of various tissues in amino acid metabolism. And we emphasize the fasting state. So, key point is all tissues need all 20 amino acids just about every day for replacing proteins, and then we have tissues that have specialized functions, like the brain has to keep synthesizing neurotransmitters. So the absence of eating, skeletal muscle is your kind of internal steak, as I like to call it. Skeletal muscle still breaks down and releases all 20 amino acids into the blood for all the tissues to use, like the liver. I mean, the liver has to keep making plasma proteins and clotting factors, and the immune system has to keep producing proteins and so on. So what the muscle does is it releases all 20 amino acids into the blood, but it takes about half or 2/3 of its branched chain amino acids and catabolizes them for its own energy needs. The branched chain amino acids are like little fatty acids. They're energy rich. So from the muscle's point of view, it's going to catabolize mainly the proteins of the contractile proteins, because it doesn't need them so much, and use them for energy. Now, in the process of catabolizing these branched chain amino acids, you got all this waste nitrogen to get rid of. So how does the muscle get rid of that? Well, what it does is it covalently attaches the nitrogen into alanine and glutamine, and then sends alanine and glutamine out into the blood. So alanine, its main fate, as I said before, is to go to the liver and become a substrate for gluconeogenesis. Glutamine goes to a lot of other tissues because it has a number of essential roles. Now, you remember from Dr. Graham's lecture on purine and pyrimidine biosynthesis. Glutamine is a nitrogen donor for purines and pyrimidines, which means it's a nitrogen donor for DNA synthesis. You need that for DNA synthesis.